And the answer is 96% of the population will be homozygous. And we get this number simply by squaring the frequency of the allele. You'll get more practice with this when we talk about mating in Module 7. Now, we don't have to do these calculations for the data in HapMap because we have the actual numbers. And so here's a larger picture. This is what you get when you click on a SNP position in the HapMap display. Instead of just seeing what we saw before, which was the frequency of the two alleles in the population. Oh, I don't need to do that. I've got it highlighted, don't I? This is what we saw in the previous slide, the frequency of the T allele and of the G allele in each of these populations. But now we also see the frequency of the diploid genotypes, people who are TT, people who are GT, people who are GG. Now, one caveat for looking at HapMap data is that much of it was determined for relatively small populations. You can see the total numbers of individuals that were tested for each, for their genotypes and their allele frequencies. So most of these numbers are actually less than 200. Most of these numbers are only about 100. What that means is that small differences between populations, between the numbers, may not represent real population differences because there's just chance effects happening when you're dealing with small numbers. So what we've done, I've given you a refresher about SNPs, thinking about polymorphisms in populations, and I've introduced you to HapMap, which is one of my favorite resources because it lets us think about natural human genetic diversity over the whole world. I've shown you how we can use HapMap to look at diversity, and what we've seen is that in different populations we find the same two alleles for each SNP. That's because we all originally descended from the same small diverse populations, but these alleles are often at different frequencies because of the different histories of our different populations. Now, coming up next, we're going to talk about using HapMap in combination with genome-wide association studies to predict the phenotypes of individuals. This is the kind of technique that 23andMe and other genotyping companies use to predict our phenotypes. I hope to see you there.